Great. So yes, thank you for coming along to this Northwest seminar in the Northwest seminar series in mathematical biology and data science. We're delighted to be joined by Guillaume Charras this week from UCL. So Guillaume's professor in cell biophysics at UCL, working on problems across scales, I'd say, from down to the cytoskeleton up to collections of cells and tissues and the mechanics across those scales. And today, you're going to be talking about rupture strength of living cell sheets. I think you said you also snuck in a bit of another topic as well.
Yeah.
So over to you.
Well, thank you very much, Carl. And it's a pleasure to be in Manchester on such a lovely day. And so, like Carl said, I'll speak about rupture strength of living sheets, but I'll also try and share some work that has to do with cell arrangement in simple cell aggregates and simple embryos. And so just so that we're all on the same page, I'll spend the first couple of slides just reminding you of epithelia and how they're organized. So epithelia are sheets of cells that line all of our internal and external organs, and they're often 1 cell thick, so about 10 microns. They're usually grow on a substrate and most often collagen. And so you could say, in a sense, that most of our tissues are composites from that point of view. And to create a mechanical continuum, the cells are linked to one another via a number of specialized intercellular junctions. And I'll speak primarily about adherence junctions that link the F-actin cytoskeleton of one cell to that of its neighbors, and desmosomes, which link the intermediate filament networks to one another. And they've got roughly the same organization in the sense that they both comprise some transmembrane proteins. Those transmembrane proteins will link cells to one another via their extracellular domains. And on the cytoplasmic side, they'll bind to the cytoskeleton, usually via the intermediary of a number of different adapter proteins. And the main physiological role of these epithelia is essentially to create a barrier between the inside and the outside. And so they'll try and restrict the passage of solutes, waste products, pathogens, etc., from the exterior of the organism into the interior of the organism.
Yeah, that's on my screen.
Yeah.
Okay.
Thank you.
Okay. And essentially these, in doing so, they constitute the first line of defense against the external environment. And one challenge to this is that you have to do all of this while functioning in the presence of mechanical stress. And so that can either be stress that's generated by the cells and the organism itself, or it can be something that is due to the normal functioning of the body. So for example, in the lung, when we breathe, the lung alveoli are distended and they're deformed by 20 to 30% at a strain rate that can reach 50 to 80% per second. In the intestine, when we digest food, you have large deformations that will take place in the intestinal epithelium. And whenever our limbs move, we'll put some strain onto our skin. So for example, if I just do this type of movement with my arm, on my shoulder blades, there are strains on the order of 60%, and I can do that really fast. So you can see that easily reach 100% per second in terms of strain rates. So it's crucial that the epithelium is able to withstand all of that and preserve that barrier between the internal and the external. So after having told you all of this, that you need a barrier, in some cases actually, and in particular in development, there are some cases when we need epithelia to break. And one nice example comes from the fruit fly and the leg imaginal disc. And in this case, the leg imaginal disc consists of 2 different epithelia, a squamous epithelium, which is called the peripodial membrane, that surrounds the leg columnar epithelium. And as the leg extends, it needs this peripodial membrane to break. And so there's some very nice work by Magali Suzanne's lab that's looked at that. And so in this movie, you can distinguish the columnar epithelium here. That's what will become the leg. And all of this stuff around is the peripodial membrane. And when the leg extends, at some stage, this peripodial membrane ruptures, and it will free the leg that can then extend. And this is something that's actually regulated from the point of view of the peripodial membrane rather than being a purely passive process. So, but despite the importance for normal physiology and for development, we know relatively little about fracture in epithelia, and that's something that we decided to try and study. And one reason we know relatively little about all of this is that it's actually difficult to study the mechanics of epithelia and to really quantify the forces that are exerted on these epithelia, because they tend to grow on a substrate. So if you deform the epithelium and substrate composite, most of the stresses tend to be borne by the substrate because it's often more rigid and thicker than the epithelium itself. So about a decade ago, we decided the best way to try and study these would be to remove the substrate altogether. And the way that we do this is that we polymerize a drop of collagen between these two test rods. Once that's polymerized, we use it as a substrate to culture cells. And once the cells cover the whole of this collagen and part of each test rod, then we can get ready to do an experiment, and we can remove the collagen with enzymatic digestion, and we're left with essentially a suspended monolayer that straddles this gap, and it's where the cells are just attached to one another via intercellular junctions. So whatever force we put on this, it's completely borne by the cytoskeleton and the intercellular junctions. We don't need to do anything complicated about deconvolving what gets borne by the substrate and what gets borne by the epithelium. So in spite of removing the substrate, the epithelium maintains some atypical basal polarity so that you still have a very clear apical side. So here we're looking at GP135 in green, which is a classic apical marker for epithelia. And that's what this looks like. So you've got one test rod here, here is the other test rod, and all of this tissue here in between is suspended and cells are just attached to one another via cell-cell junctions. And there is something like 10 or 20,000 cells within that region. So that's why I was joking earlier that what I do is more like milli-technology rather than nanotechnology. But thankfully, no one in the center has realized that so far, so I'm all good. The culture system then interfaces to a mechanical testing system, which just fits on the microscope stage. And within that, we've got the culture device, which is here. We've got a manual manipulator, which keeps a reference rod in position. And then we've got a motorized manipulator, which allows us to move the flexible rod so that we deform the monolayer, which is in magenta here. And within that, we've also got a force transducer so that we can measure the forces that we're applying at all time points. And because we're on the microscope stage, we can then film either at low magnification to get tissue information or at higher magnification if we want cellular level information. And so over the years, we've used this for a certain number of things. So for example, we've looked at the response of epithelia to a rapid decrease in their area. And what we find is that we've got some transient buckling first, and then over time, the whole tissue flattens through active processes which are guided by myosin. We've also shown that the epithelia have some spontaneous curvature, and that's due to an apical-basal gradient in myosin activity. And we've also spent some time looking at stress relaxation at very small strains. So here we were concerned with strain on the order of 30% per second, of 30%. And in those conditions, everything's dominated by the actin cytoskeleton. But so, then as an immense paradigm shift from the last one, we decided, well, what happens if we just stretch longer and longer and longer? So the experiments are super simple. All that we do is we just stretch at a constant strain rate until the tissue ruptures. And this is what it looks like. So you have the bar that's motorized and that moves. And at some stage, the tissue starts tearing, and you have a crack that propagates throughout the tissue until the whole thing ruptures. And because we can measure forces, we can then plot this as a tension versus strain diagram. And what you can see is that the tension increases up to a maximum, which corresponds to the appearance of these first cracks within the tissue, and then it decreases again. And so that allows us to define a rupture stress and a rupture strain. And the first thing that you can see from this is that the monolayers are extremely strong. You can more than triple their length before you get the first cracks. And the whole thing falls apart only once you've reached something like 5 times the initial length. Okay, but if something breaks, we usually like to know where. So if we image at much higher resolution, here you can, essentially you can see all of the cells here. And what you can see is that it's never through rupture of one of the cells that the crack progresses. Essentially what happens is that cells get peeled off from one another and at the intercellular junctions, and the crack path essentially follows all of the cell-cell junctions. So I told you in some physiological processes we get rupture, the monolayers can self-rupture. And one process that's very important in development and disease is epithelial to mesenchymal transition. And there's been some research that suggests that that can occur through a transient increase in the contractility of cells. And so to test that, what we did is that we treated these monolayers with colliculin, which is a broad spectrum phosphatase inhibitor. So in amongst other things, it also blocks myosin phosphatase. And so if you do that, what happens is that, so I'm starting to move in. What happens is that at some stage, so for a while everything is fine, and then at some stage you start getting cracks within the material, and the whole thing falls apart. So in this case, the monolayer has self-ruptured. We haven't done anything to it. We haven't deformed it. It's really just through an increase in internal contractility. And again, we can measure the tension over time. And here, what you can see is that tension increases again up to a maximum, which is when those first cracks appear. So I told you earlier that this is a bit of a messy drug. So you can also do some experiments where you combine this with a specific inhibitor of myosin contractility to show that when you do that, your tissues no longer rupture and you no longer get any increase in tension. So it's really because of the increase in contractility that you have this increase in tension and that you have the rupture that takes place. But one thing that's different between these experiments and the ones before is that the tension that we reach at rupture is 10 times lower than when I apply a ramp in deformation. And the time that it takes to get to rupture is 10 times longer. So you might ask, why is it even the same phenomenon? Yes. Yes. And so what we then did to explore that is that we reran all of our experiments with a number of different strain rates. So we go from something that's fairly fast, that's 3% per second, to something that's painfully slow at 0.1% per second. And you can, in each case, you can define a rupture stress, a rupture tension, a rupture time, etc. So if we plot the rupture tension as a function of strain rate, you can see that your rupture tension increases. It may actually plateau, but we would need a couple extra data points to say that. The rupture strain, on the contrary, decreases with strain rate, and the rupture time also decreases with strain. But this was, for us, it was a little bit surprising because our previous work, when we were looking at these small deformations, it seemed to indicate that monolayers behave like solids and more like elastic solids. And for an elastic solid, if you deform it more, you should have a higher tension. But here, if I go at very low strain rates, I deform it a lot, but it's my lowest rupture tension. So there's something there that wasn't quite clear. And it's this type of phenomenology that we spent a lot of time trying to dig into afterwards. And while I was talking, you can see this one is still going. That one is our world champion that gets to something like 10 or 11 times its initial length before you get any crack. So if you go very, 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 very slowly, you can really deform to extremes before you get any cracking in the tissue. And so you can take all of these experiments and then you can plot these in a rupture tension, rupture time diagram. And what you see is that you've got an exponential scaling between the rupture tension and the rupture time. And so essentially, if I subject my tissue to high tension, it's got a short lifetime. And if I subject it to low tension, it's got a much longer lifetime. And if I now take my colliculin experiments and I plot them in this graph, what you can see is that these actually fall close to the line that I would expect from the data where I just vary the strain rate. And so that would suggest that whether I generate my stresses passively through deformation or actively through myosin contractility, that's got the same effect on rupture and rupture time. So that in this case, they act in the same way on the tissue. Okay, and this trade-off between rupture tension and lifetime reminded us of what's known for individual bonds. So if you have an individual bond and you expose it to high force, its lifetime tends to be short. And if you expose it to low force, it tends to have a longer lifetime. And for collections of these bonds that link two rigid plates, then the rupture tension will have an exponential relationship with the rupture time. And that is very similar to what we observe in our monolayers. So this here is just the same data as there, but in a linear scale. And so what this suggests is that the rupture of my tissue at the millimeter scale is governed by the characteristics of bonds at the molecular scale. And I'll then come back to this a little bit later on. So have the tissues evolved some way of trying to resist extreme deformations? So you can get some clues into that just by looking at this curve. What you can see is that you have a change in the slope of your tension versus strain curve. And what that means is that our monolayers are strain stiffening. So in practice, it means that if I want to add any extra increment in deformation I want to add, it's going to cost me more increment in force because my tissue is becoming stiffer with strain. And one way in which I can quantify that is to take the gradient of this curve, and that gives you something that's known as the tangent modulus, which is dimensionally it's the same thing as the stiffness. And what you can see is that you go from a low stiffness regime at low strain and transition to a high stiffness regime at high strain. And that difference in stiffness is very significant. It's on the order of a 6-fold difference between the 10%, the 10-20% strain and the 100% strain. And the other thing that's interesting about this is that it's also strain rate dependent. So if I'm at low strain rate, I can't see any evidence of strain stiffening, but as I increase strain rate, the strain stiffening seems to be more and more apparent. And I'm told that that's called shear stiffening, but please don't ask me why. So we then spent quite a lot of time trying to understand what controls this shear stiffening behavior. So usually, I come from a field which is very actin-centric. So usually my response would be, well, it's actin and myosin, and there's no point in looking any further. But in this case, what we did is we inhibited myosins, we increased myosin activity, we depolymerized the whole of the actin, but you can see you still get strain stiffening. And what's more, the rupture characteristics of the material don't seem to be affected very much, so it's not effective. And it's not myosin also. And the other cytoskeletal network that we know plays a role in tissue mechanics is the keratin intermediate filament network. And this keratin intermediate filament network forms a supracellular network, and it's organized, essentially you'll have a large entanglement around the nucleus, and then it radiates out towards the cell periphery. And here I've outlined one cell, and if you follow one of these filaments, you see that if you just have the keratins in green here, it would be hard to say where one cell ends and the next cell starts. It really looks like something that's very well connected across the cell junctions. And at cell junctions, these are interfaced via a certain number of proteins. So, and I'll speak about desmoplakin in a little bit. So I just wanted to show where it is. These are the intermediate filaments. This is desmoplakin, and then it links to plakophilin, plakoglobin, and the desmosomal cotyledons. So the reason why keratins are so interesting is that we know that intermediate filaments are very extensible. When you put them in networks, they display strain stiffening. And the other thing that we know is that mutations in intermediate filaments and desmosomes give rise to a whole slew of diseases where you get fragile epithelia. And so one of these, for example, is epidermolysis bullosa. And the patients that have this disease will have cracking of their skin at each articulation. So essentially, anywhere where there's high strain on the skin, you'll tend to get cracking. So what we did is that we engineered one of the point mutations that you find in some epidermolysis bullosa patients, and that's this keratin R125C mutation. We overexpress that in cells, and I'll show you in the next slide, but that results in disruption of the keratin network. And here what I have is I've got one wild-type monolayer, and this monolayer here is made of cells that overexpress this keratin. I'm playing them side by side, it's at the same strain rate. And what you'll see is that this monolayer here cracks very early compared to the wild-type tissue. And you can, of course, you can quantify that by looking at the rupture tension and the rupture strain. And you can see that you've got lower rupture tension and lower rupture strain. So essentially, you've got a more fragile tissue. So if we also deplete desmoplakin, so that's a way of interfacing the intermediate filaments to the cell junctions, we get the same type of phenomenon. Yeah. Roughly, I guess, it will be in the tens of thousands in both cases. But you're right that this one here appears to be a little bit larger than the other one. We've actually systematically looked at that in the review process, where we were asked, is there any correlation between, the length, width, etc. of the monolayer? There doesn't seem to be. So just to check that it worked as advertised, it's a little bit washed out here, but so we did some immunostaining for keratins and desmoplakin. You can just about see here and so here you've got one cell. Around the nucleus, you've got a network that radiates out towards each of the cell junctions. Then you get a connection to the network in the other cells. And if we overexpress the keratin R125C mutation, then that network is collapsed, mostly around the nucleus. You have very few fibers that make it all the way to the cell periphery, and a lot of the keratins are aggregated. And so you remember that I started this whole, I opened this whole parenthesis because of strain stiffening. If we now look at our tangent modulus again, and we look at strain stiffening in the tissues, you can see that in the wild-type tissues, we've got this clear transition to a higher tangent modulus at high strain, but in the keratin 14 tissues, we just cannot see that. We've no longer got any strain stiffening. And similarly, if we deplete desmoplakins, we've got something similar. So the non-silencing control is in blue, and in magenta, you've got the desmoplakin shRNA. So if we disrupt the keratin filaments themselves, or if we disrupt their interfacing across cell junctions, we get the same phenotype. We don't have any more strain stiffening, and we've got more fragile tissues. So, and what you can do to just convince yourselves that these are quite different tissues is that you can place them in a rupture tension versus rupture strain diagram. Here in red is a line that tries to do a regression on the wild-type tissues, and in magenta, it's on the keratin 14 tissue. So they are phenomenologically very different as well. So overall, our experiments seem to show that living tissues are optimized to resist deformation at high strain rate, like if you have a shock, but to be very deformable at low strain rates to allow, for example, for tissue adaptation, tissue growth, etc. So this was all very nice, but all of that work so far is just done in vitro on on a cancer cell line. And so we wanted to ask if keratins play a role in vivo as well. And so we worked with Elias Barriga's lab in Portugal. And he looked at, he tried to first identify tissue in the developing frog that undergoes a large deformation. And what he realized is that when the embryos undergo axis elongation, so they elongate along the anterior-posterior axis, cells in the lateral epidermis seem to be subjected to quite a lot of deformation. So you can distinguish individual cells here in the lateral epidermis of a developing frog. And here we're looking at 10 hours or so of a movie, and you can see that the cells in this region seem to become elongated along the anterior-posterior axis. And so they then image these at high magnification. So the anterior-posterior axis is along the vertical. You can distinguish cell junctions here. These lines, the faint gray lines, are imaging artifacts, so just please pretend they don't happen. And we're looking at about 10 hours of movie here. So as time progresses, the cells become more elongated along this anterior-posterior axis. The strains are quite modest compared to what I showed you in the deformation experiments. They're more like on the order of 40%. So still large for an engineering material, but not hundreds of percent. And then what they did is that they microinjected morpholinos against keratin and desmoplakin. And I have managed to mess up my movies. Okay. So I think what I'll do, I'll just restart the presentation, because it's worth seeing them. Okay. Okay, so we have two phenotypes when we disrupt keratin and desmoplakin. We've got a mild phenotype where the cells don't really seem to elongate along the anterior-posterior axis, a little bit as if they didn't transmit stress. And then we've got a much more severe phenotype where the cells start elongating and then get a crack in the epidermis. I hope that you saw the crack before the movie stopped, but if not, I'll show you afterwards. So here, really, you know, it's the much lower strains, but we're exposing the tissue to strain for 10 hours or so. So we are compensating the low strain with the long duration of application of stress. And so again, it's probing this how long you subject to stress versus how large the stress is, or strain in this case. Okay, so I told you at the beginning or midway through that the scaling between rupture tension and rupture time was reminiscent of what happens to collections of bonds between rigid plates. But you know, epithelia and cells are not rigid plates, and so you'd ideally want to put in the rheology of those tissues in there. So I worked with Alexandre Cabla and Alessandra Bonfanti to do this. And so essentially in our experiments, what we do is we impose strain, and the deformation is going to give rise to a stress. It depends on the rheology of the material that we have. And it's that stress that will be, the bonds will sense, and that will give rise to the breakage of the bonds. So in our simulations, what we do is we have a ramp in strain, then the rheology of the material is going to give us a stress as a function of time or strain. And that stress we use as input to our stochastic bond simulations to be able to predict a rupture stress and a rupture strain. So in practice, you know, we can then ask, if we pick a rheology, we can try to find the best fit for that rheology on the rupture tension versus strain rate diagram. And here it's a linear elastic spring. You know, the fit in gray, you have the experimental data points, and in color here, you have the best fit. So here, obviously, it doesn't work very well. And then what you can do once you've calibrated your material on this curve, you can then try and predict the rupture strain versus strain rate curve. And then you can see that with this type of material, it would predict that the rupture strain should increase with strain rate, which is the contrary of what we observe in our experiments. So, okay, it's not a linear spring. You can then try to use the rheology that we had found at low strain that's based on the actin cytoskeleton, and that also doesn't work. if you try and use a strain-stiffening material, it also doesn't work. But so we really need to do something different. And what we decided to do is to have a closer look at what happens to the keratin. So at low strain, the keratins within cells tends to be quite wavy, and as if they were not subjected to stress. And then your stiffness is low, and that's probably because only the actin cytoskeleton contributes. And as you increase strain, you might iron out some of the fluctuations in these filaments. And so if you look in these cells, you start seeing some filaments that are fairly straight, and they might then contribute to bearing some load. So your stiffness now is intermediate, and at the largest of strains, you've now ironed out fluctuations in all of the possible keratins that might participate. And then in that case, you reach a plateau in your stiffness. So this is a type of strain stiffening, similar to what you would see in fibrous material. And so to implement that within our model, what we do is we use a phenomenological equivalent rheological model that consists of a spring that's in parallel with a number of branches that have a dashpot, a spring, and this is some slack. So when the slack is like this branch doesn't participate to load bearing. When we deform it a little bit, we iron out the slack in this branch, and so now we have 2 springs in parallel. If we deform more, we have 3 springs in parallel, so the whole thing stiffens over time. And then at some stage, we've ironed out all of the different slack. And because the slack is different in each branch, then all of these dashpots are out of synchrony, and so there is some relaxation in the material. And that allows us to then have a strain stiffening that's dependent upon the strain rate. So if we now take this and we fit our rupture stress as a function of strain rate, first of all, we can get a much nicer fit to the experimental data, but also our rupture strain as a function of strain rate is now fitted accurately with these type of models. Okay, so I've told you, this works, but it really relies on having dashpots in each of these branches and having this slack. So how can we try and evidence this? And what we thought we could do is that, well, maybe if we did experiments where we stretch to a deformation where we've ironed out the slack in some of the branches, and we keep it stretched, then we'll give all of these dashpots time to flow. And when we return the material back to its initial length, then what these dashpots, how much they've flowed essentially, is going to add slack to each of these branches. So what we should find is that if we do a second cycle of a stretch, now the point at which we observe strain stiffening should have changed. So what we did is we did experiments where we stretched to 75%, we kept it stretched for 20 minutes, then we return it back to its initial length. Then the next cycle we do 100%, we keep it stretched for 20 minutes, etc., etc. And then the last one, 125%. And these timings are chosen because what we know about actin is that it turns over fast. It's like 1 or 2 minutes. What we know about intermediate filaments is that they turn over slowly, more like on the hour time scale. And what's more, we know that what we're observing are not individual keratins, they're bundles of keratins. And so they're bundles of keratins that can potentially slide past one another. Okay, so all of that to say that if we now look at this diagram, the first cycle is in dark blue here, and you can see that you start strain stiffening on the order of 50%. The second cycle is in magenta, I guess. And we start strain stiffening at more on the order of 75%. And then our last cycle is in yellow, and you can see that we've got something that looks linear up to nearly 100%. So it looks like this idea that you've got something that has flowed and that has added length might be correct. If we do the experiment in tissues which have got keratin, where we overexpress the keratin 14, we really don't see that behavior at all. But if we go back to the strength of the tissue, that still leaves us with a little bit of a problem. So if I take 2 tissues, one that's linear elastic, the other one that's strain stiffened, and let's say I know everything about the tissue, I know all about the bonds, I know their number, I know all of their characteristics, I should be able to predict the rupture stress. And then if I looked at these tissues and I said, well, can I then predict the rupture strain? It should, what I would predict is that my strain stiffening materials should rupture before those that don't. But when I look at my experiments, the wild-type monolayers that strain stiffen, they rupture much later than the ones that don't strain stiffen. So I have a little bit of a problem here. And one explanation for that problem might be due to catch bond properties for the desmosomal coherence. So a catch bond is a bond whose lifetime is short when it's not exposed to any force. Then when you expose it to force, its lifetime will reach an optimum. And then when you exceed that force, the lifetime decreases. So what might happen is that at low strain, I'm not putting any force on the desmosomes. So the desmosomal coherence might have a short lifetime. It would make a small contribution to adhesion. And so that might be why I have a low rupture tension. Now, high strain, I'm putting some force on the desmosomes. Their lifetime might become longer, make a larger contribution to adhesion. And so that might be why you've got a larger rupture tension. And so when I stretch my tissues, I'm doing two things. I'm strain stiffening them, but I'm also increasing the adhesion between cells. So in terms of, and since I'm increasing the adhesion between cells, my rupture tension prediction changes, and now I can have a rupture strain that would be larger than in my non-strain stiffening tissues. So in terms of evidence, if we look at the transcripts for ecadherin, the quantity is equivalent to that you have for the desmosomal coherens, roughly speaking at least. And desmosomal coherens have been shown to have similar force lifetime behaviors as ecadherins. What is missing is that they haven't been shown to have this type of behavior. Okay, and so what we can do is we can use our model junctions to try and probe the differences in adhesion. And the way that we use these is that we've got, essentially what we have is we've got, we're modeling a collection of bonds that's subjected to a certain amount of force. It's subjected to a certain amount of force. And then over time, we monitor the state of each of these bonds. So here we've got the different bonds, and when they're broken, they're in black. And this is time, and what you can see is that, you know, over time, you'll increase the number of broken bonds, and at some stage, all of the bonds are broken, and that's what we define as the rupture stress. Okay, and our model is parameterized by now our on and off rate for the bonds. There's a force dependency in the model, and then there's the number of bonds in there. And so if you change the number of bonds, or if you change the force dependency, you decrease the rupture stress. And so as a first step, what we did is we assumed that the rupture characteristics under force of desmosomal coherence and ecoherence was the same. And we assumed that only the number of bonds could change. And so what we did is we took our experiments, our tension versus time experiments, and we subject our models to these experimental curves. And then we adjust the number of bonds until half of our experimental runs rupture before the rupture stress for the experiment and half of them afterwards. And that means we've reached roughly the right number of adhesions. And so when we do this, what we find is that there doesn't seem to be any difference in adhesion in the wild type tissues if we go at different strain rates, and that's reassuring. If we look at the keratin 14s, they seem, the monolayers with keratin 14 overexpression, they seem to have much less adhesion than the wild type tissues. And then the tissues that we treated with colliculin that rupture through increasing contractility but no deformation also seem to have lower adhesion. And that might be because we're not deforming them, we're not soliciting the desmosomes. Okay, so in conclusion to this part, actomyosin dominates mechanics at low strain. The intermediate filaments seem to dominate at high strain, and they underlie strain stiffening. The deformation leads to stiffening and also an increase in intercellular adhesion. And essentially, the message is that, you know, the tissue rupture at the millimeter scale is controlled by the molecular scale dynamics of bonds. And of course, there's quite a lot of, I think, interesting open questions like, we sort of scratched the surface on what underlies the dependency on loading history. And that would be interesting to look at more in detail. But also, we've now got two cytoskeletal networks that bear load, and it's not clear how you share the load between these networks. So I've been a little bit slow, so I'll try and do this next part fast, but we'll see how well I manage. I'll aim to be finished in 10 minutes.
Yeah.
Okay. So the other thing that I wanted to talk to you about is essentially we've been spending some time trying to understand how mechanical differences between single cells integrate in a very simple cell aggregate to give rise to shape. And that these complex shapes can actually occur in pretty simple aggregates. So what we looked at was C. elegans, and in this movie, the embryo is inside its eggshell. It starts from one cell. You have an asymmetric division. This is the larger of the cell called AB. And then AB divides symmetrically to give ABA and ABP. And then P1 divides asymmetrically again. And what you can see is you've got this aggregate of cells here at the rear. And then this cell here seems to poke out a little bit and be excluded. That's called P2. So one reason could just be that you're in a confined environment or in an eggshell. But actually, you can take the embryos out of the eggshell. And in this case, what you can see is that it's very clear that these three cells here, which are the two AB cells, EMS, and this is P2, these three cells form a very compact aggregate, and this P2 cell is excluded from the others. And, 4 cells seem like something you could probably try and do experimentally. So we tried to look into the mechanical difference between these to see if we could learn something. So for small aggregates of cells at steady state, one of the theories that's worked really well comes from foam mechanics. And in this case, all of the arrangement of cells is governed by the surface tension. So if you have two cells that interact, the angle of contact here is controlled by the surface tension at the free membrane and the surface tension at the cell-cell contact. And there's actually some theoretical predictions from the Young-Dupré equation that tell you what this contact angle should be. And if the contact angle changes, for example, if it increases, it could be because the cortical tension here has increased or because the junctional tension has decreased. But if you don't do an experiment, you can't differentiate these two. On a molecular level, the tension at the free surfaces, the cortical tension, is controlled by myosin and actin. And then at cell-cell contacts, you've got cadherins that come in and play a role. And the cadherins can have two contributions. They can either have an indirect contribution via signaling, or they can have a direct contribution via the adhesion. And I'll skip this. And if you want to convince yourself that there are some changes in mechanics, what you can do is you can look at your embryo at different time points, and you can try and measure the angles of contact between cells. So you can measure the angles of contact between cells within the same embryo. So you can see right away that this angle and that angle, they're not the same. So there must be differences between the pairs of cells in there. And then these cells here, they're the progeny of these cells. And you can see again that the angle of contact isn't the same. And one of the sources of these differences could be mechanics. And in this first asymmetric division, you've got an asymmetric partitioning of myosin and actin. So for example, if you look at the myosin, you can see that the two AB cells, which are the daughters of AB, which was the larger of the two cells, they've got most of the myosin, whereas EMS and P2 seem to have far less myosin. So essentially, we set off from there and we decided to try and see what we could do in terms of mechanics. So to understand the arrangement of the embryo, we need to measure the cortical tensions and the tension at cell-cell contacts. So for cortical tensions, we use atomic force microscopy to measure these. And essentially what we do is we bring a microfabricated cantilever on top of a cell, and we can measure the force that we apply for a certain deformation. That's why I'm in the Center for Nanotechnology. So this is what it looks like in profile. So you've got the cantilever, this is a cell, that's the substrate. And from above, you've got the cantilever, the cell, and here you have the rest of the embryo. So it's essentially, it's the same experiment that you do if you've got a football and you want to see if it's inflated. You press against it. You know what force you're putting, and you have an idea of how much you're deforming. And it's exactly the same thing you do there. Yeah. Yes, you analyze it in that way. So when you press on the cell in red here, initially you're out of contact, then you come into contact with the cell, you get a peak force, and then that decays over 20 to 30 seconds. That decay is due, we think, to turnover of the actin cytoskeleton, and then it reaches a plateau, and from the value of force at this plateau and the geometry of the cell, you can estimate the cortical tension. If you do the same thing for the eggshell, what you can see is it doesn't relax at all. So it's elastic one being viscoelastic, and it's actually much stiffer than the cell. And that's useful later on. So with that, you can draw a panorama of the surface tension in the cells of the embryo. And what you can see is that cells of the AB lineage have got a higher surface tension than cells of the P lineage, so P1 and its descendants, EMS and P2. The surface tension depends on the cell cycle. So if you block cells in metaphase, they've got a much larger surface tension than when they're in interphase. And then if you block myosin contractility, they've got a lower surface tension. So it mirrors what we know about mammalian cells. But I told you all of this is dynamic. So what you can also do is you can just leave the cantilever in contact with the cell over time. And then you can image progressions through the cell cycle looking at histone markers. And so here I'm looking at the surface tension over time. And what we can see is that the AB cells increase their tension over time. EMS actually starts with a very low surface tension, then it increases up to a point where it's nearly the same as the AB cells. And so that might be why these three cells in the rear compact closer together. And then P2 stays low in terms of surface tension. So we wanted to ask where those dynamics come from. And here I come back to being actomyosin centric. So myosin is a good candidate for that. So what we did is we used embryos that express a genetic replacement for myosin 2 with a GFP fusion. And so we can then segment the amount of GFP myosin that we have at the cortex. And then we can plot the surface tension that we measure by AFM as a function of the myosin intensity. And we've got a scaling that seems fairly convincing. It's not perfect, and I'm happy to discuss this with anyone afterwards. But what it does do is it gives us a general order of magnitude for the surface tension as a function of the amount of myosin that you have at the cell surface. So one of the techniques that people use when they look at arrangement of cells in aggregates is they'll use Bayesian inference to have an idea of the relative surface tensions in there. And that relies on two assumptions. One is that cells behave a little bit like foams, and the other one that you're in quasi-static. Based on that, we worked with Hervé Turlier in Collège de France, and what they did is they segmented our movies, found the angles of contact. Then if you have that for each and every cell, you've got enough equations to solve the whole system with one reference tension. And so you can then use all of these relative tensions to put into simulations to get some information about the shape of the embryo. So as a first step, it looks very good. It looks relatively similar to what you have. And you can see that over time, you've got some compaction in the rear here, but also you can see that this P2 cell does come a little bit closer to the other cells over time. And what we can then, the issue with these type of measurements is often linking them in time, because it's all relative to a reference. And if you don't know how that reference evolves in time, then you can't link those snapshots. But what we can do is we can use our relationship between myosin intensity and tension to use that as a reference, whose temporal evolution we would know. And if we do that, we can get an estimate for the absolute surface tensions that are in the embryo. So what you can see is that P2 essentially sticks out of this group because the tension at its cell-cell contact is larger than the tension at its free surface, whereas these make a compact group because the tension at free surface is larger than the tension at cell-cell contact. And you can, if you make the assumption that everything stays the same in terms of the relationship between myosin and tension within the eggshell, then you can do the same type of predictions in the eggshell. And I mean, essentially, it's roughly the same. So early on, you've got higher surface tensions in the free surfaces compared to cell-cell junctions. You've got a higher cell-cell contact tension here compared to the cortical tension in P2. Maybe the only difference is that over time, P2 does contact more with that aggregate later on.
The other one is characterize over time, and then this one looks kind of less symmetric over time.
You mean in terms of the arrangement of these cells? Yeah. Yes, you're right. You're right, but you have an extra junction in here. So this junction here doesn't exist when we remove the confinement, because it's when P1 divides, it's got no other way to go and it needs to come into contact with P2. That could be an explanation. I don't know if it's correct. But I just wanted to get to a last point. So we then wanted to ask if we can find some information about tension at cell-cell contacts. So what we did is the first step is we said, well, okay, let's just assume that myosin is also a good predictor. And we can plot the surface tension that we get from geometry versus the surface tension that we infer just in fluorescence. What you can see is that for the external surfaces, it's okay. But for the cell-cell contacts, it's really not great when we overestimate. And that's because at cell-cell contacts, there's another contribution to surface tension that comes from intercellular adhesion in the form of keratin. So what you can do is you can play the same game. You can take embryos which have got a replacement for keratin with a GFP fusion, and then you can plot this extra tension that we predict from myosin. You can plot it as a function of the keratin intensity. And what you find is that you've got a nonlinear relationship between the residual surface tension and the keratin fluorescence intensity. And that's interesting in the sense that it quantitatively, I think, tells us something about how keratin cell-cell adhesion directly contributes to the surface tension at contacts. Okay, and so we've gone through all of these, and I would like, before I finish, I'd like to thank the people who actually did the work. So Julia Duque here did most of the experiments on rupture, and she had some help from Emily Sitcha, Jonathan Fouchard, Andrew Harris is from this. This is Alessandra Bonfanti, who did most of the modeling. Ines Barriga and Sarah Azenia did the Xenophus work. Kazunori Yamamoto did most of the work on the C. elegans, and it was a collaboration with Hervé Tualier's lab in Paris. I'd like to thank you for your attention.